And our next speaker is Stephen Lomeli Gonzalez, and he's from San Francisco State BUILD, and we'll be talking about the role of March 1 in an experimental model of multiple sclerosis.
Right. Hello, everybody.
You're good to go.
Good afternoon. My name is Stephen Lomeli Gonzalez. I'm graduating with degrees in kinesiology and biology from San Francisco State University. I'm also an undergraduate researcher in the lab at Young-Suk Shin over at UCSF. And I'm going to be presenting a couple of experiments involving the role of March 1 in an experimental model of multiple sclerosis, or MS. Multiple sclerosis is an autoimmune disease affecting the central nervous system. It currently affects more than 3 million people worldwide with no cure. Individuals suffering from multiple sclerosis experience compromised myelin sheath as well as axons. And when individuals are suffering from multiple sclerosis, they suffer from symptoms that include blurry vision, dysfunctional bladder, muscle weakness, tremors, and in severe cases, total paralysis, and even death. Research has implicated T cells as a mediator of the disease. However, the mechanism is still unknown. Our lab has long been interested in the dynamics between T cells and their activation through antigen-presenting cells. March1 is a protein that's specifically expressed by antigen-presenting cells and controls the expression of these T cells that are essential for the activation and multiple sclerosis. It led us to wonder the role of March1's involvement in MS by supporting the activation of myelin-specific cytokine-producing T cells. I adopted a well-established animal model of multiple sclerosis called experimental autoimmune encephalomyelitis to test my hypothesis because it mimics the pathology of multiple sclerosis. What I did first is I immunized 2 sets of mice, wild type and March1 knockout. So the March1 knockout mice were deficient in the expression of March1. First immunized them with myelin peptide, seen here as MOG3555, as well as an adjuvant. And then on day 0 and day 2, I also injected them with pertussis toxin, a known virulent factor that compromises the blood-brain barrier. From then on out, I would clinically score them and weigh them until the termination of the project. The score was indicative of how far along the disease progression they were, experiencing symptoms such as tail paralysis, hind limb, and forelimb paralysis. Now let me walk you through a visual representation of the data. The first graph here, seen in red, represents the March1 knockout mice, and in black, wild type. Upon induction of the disease, we see here that they both experience weight loss. However, the March1 knockout mice quickly recover, gaining over 100% of their weight back. However, the wild type mice weren't so fortunate. They lost over 20% of their weight, and when you're a little mouse that only weighs 20 some odd grams, that's a very significant portion of your weight. The clinical score also extremely severe between the two groups, where the wild type group experienced the most severity. And pictured here in the graphs are the production of these inflammatory T cells as well as their inflammatory markers, GM-CSF, interferon gamma, and IL-17, all implicated in deriving multiple sclerosis. It ended up supporting our hypothesis that March1 is necessary for the development of EAE. But where exactly? I then used myelin-specific transgenic mice to determine the role of March1 in developing these myelin-specific T cells. In a protocol similar to the last, I adoptively transferred these 2D2 cells that were very specific to myelin. I then injected these 2D2 cells into control, wild type, and March1 knockouts. But I only immunized the wild type and knockout mice. And instead of carrying the project full term, I harvested the draining lymph node, and then I monitored for the expansion of these myelin-specific cytokine-producing T cells using a component of the T cell receptor seen here as B-alpha 3.2, as well as a congenic marker CD45.1. Pictured here are representative flow cytometry plots of the wild type and March1 knockout group. The production of these inflammatory T cells, as well as their cytokine, was very similar. And to truly appreciate this experiment, seen here in these graphs, we notice that these 2D2 cells are present However, they did not expand because they did not receive the same treatment that the wild type in the March 1 knockout group. We concluded that there was no defect in March 1 knockout mice in developing these monospecific cytokine producing T cells. And that brings me to our future directions. I'm going to be examining the role of CD11C dendritic cells using the Cree-Loxley system, as well as inducting Tregs. And I hope to do this under the guidance of Dr. Shin as well as Carlos Castaneos, under the support of NIH Diversity Funding Grant. And with that, I'd like to thank SF BUILD for their continuous support these last 2 years, SFSU, UCSF, the NIH, and the SFSU Brazilian Student Striking Club. Carlos Castaneos was and has been my research mentor and initially brought me to the lab. He's been extremely instrumental in my development of science. He's spent so many hours with me, and I am extremely grateful, as well as Dr. Shin, who has also mentored me and has spent countless hours with me and preparing for these experiments. And with that, I'll open it up to any questions.
Thank you, Stephen. That's an excellent talk, understanding how March 1 is affecting autoimmune diseases of CNS. Are there questions from our chat?
I also want to thank all of my friends and family who ended up showing up to this talk. I realize that this type of science may not be everyone's forte, but I'm so glad that everybody is out here supporting science during these pandemic times.
That's a great sentiment, Stephen. So I'm going to give a really general question. How would you explain this to like my grandmother in 30 seconds? What is your study doing?
So if I was to explain this to someone who really wasn't in the know, I would let them know that first we induce the disease in an animal model, because obviously we can induce it in a human to be able to test. And then from there on out, we looked at the differences between the two based on their genetic makeup. And then from there, we are able to then modify the treatment or the protocol.
Thank you so much. I did that for my own benefit, but perhaps others on the listening on the call and also would benefit from it. Are there other questions? It seems your hypothesis of March 1 involvement in the disease may be confirmed. I was wondering if you could elaborate on the implications that could have for future understanding of the disease and possible treatment, and that comes from Levi Nipple.
I'm sorry, can you repeat the question one more time?
Yeah, definitely. It seems your hypothesis of March 1's involvement in the disease may be confirmed. I was wondering if you could elaborate on the implications that could have for future understanding of the disease and possible treatment.
Yeah, well, initially, like all translational science, starts off with these sets of experiments. And right now, it seems that with these sets of experiments, that March 1 ends up being the factor that makes mice resistant. We're resistant to developing AE, or in humans, it would be multiple sclerosis. So down the line, once more research is done, perhaps inhibitors that may end up helping the disease until we ultimately find mechanisms for how these inflammatory T cells end up degrading our axons and our nerves and causing the disease. Thanks for the question, Levi.
Cesar has a question for you as well. What was your sample size, and how was the environment controlled for your samples, for both the control and the knockouts?
Gotcha. Great question, Cesar. Well, I am blinded to the genetic background of the mice, and they are housed in our animal facilities over at UCSF. And what was the question on the chat? There we go. The sample size was, for each experiment was over 10 mice per group, so 10 wild type or over 10 March 1 knockout mice.
And one last question. Everyone basically on the chat says, excellent job, and I'd like to concur with that as well. What drew you to this type of research and this topic in general? Do you have a connection, or was there an interest in working with mice or working in autoimmune diseases?
Well, initially I had experience because of a course I took over at San Francisco State University. And through SF BUILD, one of the assistant directors, Yasmin, ended up setting up sessions with graduate students that were looking for mentees. And so I was very fortunate to hit off with Carlos, who is a fifth-year PhD candidate, extremely brilliant. And he took me under his wing and taught me everything I know. I mean, from the beginning of how to induce the disease, how to look at papers. I mean, I can't explain to you how instrumental he has been in the development of being a scientist, because I was extremely new to research. And just like all things, I still needed, and I'm still developing now, but I personally needed to be shown how to be a scientist, how to act like a scientist, how to think of it, because it isn't something that came naturally to me. So everything you see here is a representation of the mentors that have put in work with me. And Young Shin and Carlos, I can't be more grateful to them for allowing me the opportunity to be in their lab.
One last question. What are your plans for the future in education? And when are you going to start being a mentor to the next group of scientists?
Great question. So I am currently a member, and I have been a member. SF BUILD over at San Francisco State University has a program called the Near Peer Mentorship Program, where we take in underclassmen that happen to be science majors. We guide them through the process whenever they need. I'm also very fortunate enough to be an EOP mentor, Educational Opportunity Programs mentor. And I, again, help individuals who are on the science path or maybe questioning it. So I am currently doing some mentoring, but as far as my plans for the future, just like most of the individuals graduating, I had plans to stay in the lab of Dr. Shin and continue researching, build up my experience, and then apply to PhD programs the following year.
Excellent job, Stephen. If people are in the audience, it would be clapping, but since we're all online, you're going to just have to look at the chat box.
Well done, Stephen. Way to go. Way to rep SFSU.
I think there is a question. There is one question. Is your group planning on publishing the results of this study?
I think there have been some hiccups, and we still have some more experiments to do, but that is something that I really want to do, and I really want to learn how to write a manuscript and develop that process. Right now, I have been very fortunate to learn how to conduct experiments, how to perform them by myself, along with help of other individuals from my lab. But yeah, that's something definitely that I really want to look into. It'd be great to be able to put my name out there and contribute to the science community.
Thank you, Stephen. Well, your name's out there with us, and we look forward to hearing about your next steps. Excellent job in representing SF BUILD.
Thank you again for the opportunity. And thank you again, everybody who tuned in.
And don't go away, because there's still amazing presentations coming up. In fact, we're going to transition to our next speaker, Constantina Orselli from California State University Northridge CSUN BUILD.